Welcome! Today we will walk through the process of estimating the organic carbon content in soil, a crucial parameter for soil health. Organic carbon is vital for soil fertility, structure, water retention, and overall ecosystem function. We will be using the Walkley-Black method, which involves chemical digestion and titration to quantify organic carbon. The soil is digested with potassium dichromate and concentrated sulfuric acid. The heat from the sulfuric acid speeds up the oxidation of organic matter. The amount of excess unreduced dichromate is then determined by titrating the solution with a standard ferrous sulfate solution, using orthophenanthroline as an indicator. Now let's look at the necessary equipment and chemicals for this analysis. You'll need common lab equipment, including a 500 milliliter conical flask, a pipette, a burette, and an analytical balance for precise measurements. The key reagents include 1 normal potassium dichromate, 0.5 normal ferrous sulfate or ferrous ammonium sulfate, concentrated sulfuric acid, and ferroin indicator. Here is the detailed procedure to follow for accurate results. First, weigh exactly 1.0 gram of soil sample into a dry conical flask. Next, add 10 milliliters of 1 normal potassium dichromate and 20 milliliters of concentrated sulfuric acid. Mix thoroughly and allow the reaction to proceed for 30 minutes on an asbestos sheet for safe heat dissipation. Dilute the reaction mixture with 200 milliliters of water, then add 7 to 8 drops of ferroin indicator. Titrate the solution with standard 0.5 normal ferrous ammonium sulfate solution. The endpoint is reached when the color changes from greenish blue to reddish brown. Crucially, a blank titration without the soil sample must be run simultaneously to ensure accurate calculation of the amount of dichromate consumed by the organic matter. Finally, we move to the calculations and interpretation of the results. Use the formula provided to calculate the percentage of organic carbon based on the difference between the blank and sample titration volumes. The result is further used to estimate organic matter and nitrogen content. The organic carbon percentage is classified into tiers, from very low to very high, which guides recommendations for soil amendments like manure or biocompost. Accurate estimation of organic carbon is foundational for effective soil management and sustainable agriculture. Follow these steps for reliable analysis.